Alright, so here we have the DNA represented in Minecraft. What we have here are the two strands of DNA that are being separated by the helicase. That is the job of the helicase to separate the strands of DNA. In this case, the helicase is represented by the cows kind of like digging into the DNA strands, and within the DNA strands, we see the hydrogen bonds representing with the like gel cell like chains, and also the nucleotides and like A, G, C, and T represented in different colors. Now we have polymerase, which is here. So with the DNA polymerase, these enzymes are essential for the DNA replication process. They usually work in groups of two or more. That's why we have many different pistons working with each other. And they help turn a single DNA strand into two identical strands. In this process, the enzymes read the information from existing DNA strands and add nucleotides in the direction of three to complete the two strands. Next, we have primase. So, DNA primase is an enzyme that helps the polymerases during DNA replication process. Primase synthesize the primers, which are short RNA sequences that also serve as starting strand of DNA. The nucleotides that the polymerases add are attached to the primers, as we can see here. Uh, Another enzyme is the ligase. Here we see that DNA ligase is essential for sealing breaks in the DNA strand to maintain stability. That's why we see the pistons, adding the blocks there. It plays a key role in DNA replication by joining Okazaki fragments on the lagging strand, ensuring a continuous strand. Additionally, it is involved in DNA repair when strands are broken. One example of DNA ligase is ligase IV, which is important for repairing double strand breaks. Um, Now here we're going to talk about the breaks of the DNA. So we have the double helix here, but then it breaks over here. Um, we have the two different directions where it runs in opposite directions shown by the arrows. Here's one of the arrows and the other one is there. One strand is positioned in the 5 to 3 direction, while the other to the 3 to 5 direction. During replication, DNA polymerase can only add new nucleotides in the 5 to 3 direction. This causes the leading strands to be synthesized continuously towards a replication fork, while the lagging strand is synthesized in short Okazaki fragments moving away from the fork. These fragments are later joined by DNA ligase to create a continuous strand. Now, to show how DNA is connected with chromosomes, here we have a Y chromosome, and we see the same material that we made with DNA tightly packed for the chromosome. And how it's connected is that it stores genetic information. In eukaryotic cells, DNA wraps around proteins called histones, forming compact structures known as chromatin. During cell division, chromatin condenses further into visible chromosome, ensuring that DNA is efficiently copied and passed to new cells. Each chromosome contains genes, which are specific segments of DNA that code for proteins and determine traits. That's it for our DNA project. Thank you.